All right, here are your video notes on RNA, the genetic code, and protein synthesis. So the genetic code itself is that DNA and RNA are read 3 bases at a time. These are something called codons. So what is a codon? It's a 3-base sequence that codes for specific amino acids, and there's actually 64 possible codons. So let's do a quick review. Amino acids make proteins. There are how many different amino acids? 20. So when we look at the breakdown of codons, we take a sequence of RNA and then we separate it into codons, or basically every 3 bases you do a division. So you could do lines like this, which ultimately end up looking something like that. Later on, we're going to use these codons to identify the amino acids that correspond to them. Here's a look at the structure of RNA. The first thing you should notice is that RNA is single-stranded. Remember, DNA is a double helix, so it has 2 strands. RNA is single-stranded. Another thing you should notice is that there's no more thymine. This time we have a base called uracil. So let's take a look at why it's different. First of all, the name, right? RNA is ribonucleic acid, so RNA has the sugar ribose instead of deoxyribose. Like we talked about, it has 3 of the same bases, so A, G, and C, but this time we have uracil, which is a pyrimidine, and they still pair as follows. So instead of pairing with T, A is now going to pair with U, and then G pairs with C, which is the same as DNA. Like I said, usually single-stranded, and a bonus perk is that RNA can leave the nucleus, DNA cannot. There are 3 different types of RNA that we'll talk about. There's messenger RNA, or mRNA, and this just carries the genetic code, usually from the nucleus to the ribosome. There's ribosomal RNA, or rRNA, and this joins mRNA to tRNA in the ribosome. And then there's transfer RNA, or tRNA, and this transports amino acids to ultimately make those polypeptide chains, or the proteins. Here's a better look at just those different types of RNA. So you see tRNA is incoming, it has amino acids connected to it. mRNA is here, it's made from DNA, and then rRNA is part of the ribosome. The important thing to also remember is where it all happens. So like I mentioned, mRNA can move from the nucleus to the ribosome. This is why mRNA is used instead of DNA. Then rRNA is only in the ribosome, and tRNA goes from the ribosome to the cytoplasm. The first step of protein synthesis is something called transcription, which is making DNA into RNA. So when we take a closer look, this is when RNA polymerase is going to bind to DNA and it makes a complementary strand of mRNA. Like we mentioned, C and G still pair, but now A and U pair. And why is mRNA even used? mRNA can leave the nucleus, DNA cannot. There's also something you need to know called introns and extrons. So sometimes the code is interrupted by sequences that aren't going to be part of the final mRNA sequence. These intervening sequences are called introns, which are eventually cut out by enzymes. The RNA that actually codes for future proteins are called exons, and these are kept and eventually spliced into a single strand of mRNA. So take a look at this image. You may want to pause it here and just take a brief look at what's going on there. So when we do transcription, it's similar to DNA replication, except the bases are going to pair differently. So like we said, A now pairs with U, and G pairs with C still. So let's practice. So A goes with U, G goes with C, C goes with G. T goes with A, C with G, C with G, G goes with C, A goes with U, T goes with A, G goes with C, C with G, A with U, T with A with U, C with G, T with A, T with A, G with C, and G, G, U. Running out of room there. All right, take a minute, pause it, and go ahead and work through the second strand. All right, hopefully you got something like what's below. The next step in the process of making a protein is called translation, and this is using mRNA to make amino acids. mRNA to make amino acids. So there's something you need to be aware of called the Beadle and Tatum hypothesis. So initially, scientists actually thought that it was one gene, one enzyme. And this is the theory that each gene produces a single enzyme. Now, this was in 1941. Later on, about 15 years later, they actually figured out that it was one gene, one polypeptide. So it was modified because polypeptide chains were actually what genes coded for, and this specified a protein product. So you should know, important part here, translation is the coding of mRNA into polypeptide chains, which are also known as protein. So when we talk about the specifics of translation, so after that transcription happens, mRNA is going to leave the nucleus. From there, it attaches to the ribosome, and then tRNA also plays a role because it brings the amino acid to the ribosome. It carries these in the form of anticodons, so they're going to pair with the correct codon. And those amino acids form peptide bonds, and they're held together, and they eventually form something called a polypeptide chain or a protein. There's also some tRNA binding sites that you need to be aware of. So there's the A, the P, and the E site. And it's kind of different than what you would expect. You don't read it from right to left, you go from left to right. Or I'm sorry, you don't go from left to right, you go from right to left. So if you see here, we have A, the aminoacyl site. So this is where that tRNA is going to come in and bond. Then it moves to the P site or the peptidyl site. That's where the tRNA brings that amino acid that connects to the protein. Then it moves to the exit site. So now we have an uncharged tRNA molecule which doesn't have an amino acid attached. And then that tRNA is going to exit the ribosome. So when we do translation, this is just showing the elongation of that polypeptide chain. So as you can see here, we have numbers 1, 2, 3, and 4. So this is in the P site. This is in the A site. Okay, so 4, now eventually when we go over, 4 is going to move to the P site, and as you notice, 3 popped off of this tRNA molecule, and now 4 is attached. And then same thing, we show it here, we show that tRNA leaving without the amino acid attached. Here's another better look at translation. This also shows transcription as well. If I were you, I'd pause it here and either sketch this in your notes or screenshot it. This is a really important diagram. So when we review codon practice, we need to go over some basics. So first of all, this is a codon table, and we will use these a lot in class. You will have one of these to use on your test as well. But you do need to know the start codon. So this is how the process of translation starts. And the start codon, the only one is AUG. And it's way easier for you if you just memorize that. There are 3 stop codons, which you can see in the chart here. And they're also listed there for you. So any of those are going to signal stop, which is the stopping of the protein. So let's do some translation practice. So you're going to need a codon table to do this. So I would pause, and you can look one up on Google or on your phone and then try to do this. So basically, I'll show you. You're going to read it from left to top to right. So for example, let's go back and look. So AUG we know is start. CCG. So C and C, you find out where they pair. Whoopsie. All right, so you find out where C and C pair. So you can see that they're in this area, and then our third letter is G, so CCG is proline. So then we would write PRO. All right, go ahead and pause the video and then try to solve the rest of these on your own. All right, so here are the solved translation practices. Now you may notice a couple things. First of all, notice how I X'd out CGC. That's because this codes for stop. So when we hit stop, we don't keep going. Same thing with the bottom one here. I crossed out CCC because that's not a start codon. So you can't start until you get a start, just like you can't go through an intersection until you get a green light. And then this one also stopped early too. So you only want to code from starts to stops. All right, here's another image that just shows that. You may want to pause here and look this over and kind of practice reading it to yourself. So something that comes up frequently in this unit is the term central dogma. So what this means is just basically how DNA is ultimately made into a protein. So as you see here, DNA can recycle itself over and over. through replication. But DNA is also used to make RNA through transcription. RNA is used to make protein through translation, and that's how we get a protein from DNA. There can be some problems though, right? So what happens when things go wrong? This is something called mutations. As you can see, this poor duck here has 4 feet. Could be a problem. So what is a mutation? So a mutation is just a permanent change in the cell's DNA. And certain things can cause it. It can occur spontaneously, or it can occur from chemicals and radiation. There are 2 main types that you need to be aware of. There's a gene mutation, which is just changes in a single gene. And then there's chromosomal mutations, which are changes in an entire chromosome. And I'm sure you can guess which one is worse. Before we go on though, let's do a quick review of the cell types. So 2 terms you should have heard by now are somatic cells and sex cells. So somatic cells are body cells and sex cells are reproductive cells. The thing that you need to be aware of is what is passed down and what isn't. So mutations in somatic cells or your body cells are what to the next generation? The answer is not passed. If you get a cut on your arm, your child's not going to be born with that same scar on their arm from where you cut yourself. Whereas sex cell mutations are passed down to the offspring. So if you have an issue with your sex cell, that's what's used to make your child. So of course they're going to have this mutation. So when we look at gene mutations, there's 2 major ones. There's point mutations and frame shift mutations. So point mutation is just that. It occurs at a single point in that chain, aka only one nitrogenous base is being changed. This is something like a substitution. So if you think about sports, for example, if someone comes out, even let's say they're injured, someone gets injured on the field, you don't keep playing with one less player. You put a different player in. So this is a substitution. There's also frame shift. And when you think of frame shift, I want you to think of a reading frame. So this is a mutation that occurs when a nucleotide, a base, A, T, G, or C, is either inserted or deleted. And as a result, it changes the reading frame. So if you're reading every 3 letters, if all of a sudden you have an extra letter, it's going to change everything after that because now it's reading a different 3 letters every time. And this is super simple to think about. An insertion, you're adding, a deletion, you're removing. So here's just another look at gene mutations. So it's showing the difference and how that can be affected. And this especially shows you how that frame shift can really impact. So if this letter G is taken out, look at the amino acid sequence. That met, glu, ala, gly now becomes met, lys, his, and we have 2 random bases all by themselves. That's going to cause major issues in an organism. Here's another thing of just different types of mutations. You may want to pause this. Some of these diseases may sound familiar to you. And then we have our chromosomal mutations. So this is a change in the number or the structure of the chromosome, and there's 4 different kinds. First of all, deletion. This one's easy, right? We lose something. So if you look at these bases here, we see, oh, look, that B is gone over here, so it's clearly a deletion. Then you have a duplication. So a duplication, you can duplicate one letter or all of it. It just depends. In this situation, that B has been duplicated, so we have 2 B's. Inversion is a flip. So instead of it being A, B, C, D, E, F, the E, D, C, and B all switched. So these 2 were supposed to be on the other sides of the chromosome. And finally, something called a translocation. So this is when the chromosomes are essentially crossing what they have. So it should have been A, B, C, D, E, F, and G, H, I, J, K, L, but now we have A, B, C, J, K, L. Oh, that's not right. And then G, H, I, D, E, F. So this is also going to cause some major issues. And then here's just a look at some practice. So pause this here and work through it. All you're going to do is just give the mRNA sequence. All right, so if you go ahead and check your work, you'll see the substitution was just a letter switched out. So instead of being GCAUG, we have GAAUG. Insertion, 1 base was added. Forgive my terrible handwriting. So we have GCUACC. Now we have GCUUACC. And finally, the deletion, GCUACC. Now we just have GUACC. And that's it for your notes.